Hi. Okay, let's talk about a few more types of evolutionary adaptations and also the hormones that are involved with the digestive system. So, as you can imagine, there are a lot of dental adaptations that go along with different diets. That's one of the ways that scientists determine what kind of a diet an ancient or extinct animal consumed is by looking at their teeth. Mammals, again, are generally the only vertebrates that have multiple kinds of teeth in one mouth. Generally, for other vertebrates, fish, reptiles, all the teeth will be the same or almost the same. And of course, like everything else in biology, there's a few exceptions to that. For example, venomous snakes have fangs that can eject venom that are obviously a different size and shape from the rest of the teeth in the mouth. So here's an example of a carnivore, a herbivore, and us, an omnivore. And you can see that there's different specialized teeth. Carnivores have, even their molars are specialized for tearing and are generally pointy, and we don't have that. Herbivores have specialized teeth for grinding and also in the front for breaking off plant material. And our teeth are very much like the rest of the primates. The biggest difference between our teeth and other primates is a large reduction in the size of the canine teeth. Which you might wonder, why would other primates have very large canine teeth, which are generally associated with being a carnivore, if primates are all omnivores? And it's thought that the large canine teeth of, for example, chimpanzees and gorillas are used as threat displays to other individuals of the same species and aren't used for catching prey per se, like they're used for in carnivores. So as I mentioned a little bit in the last video, there are stomach and intestinal differences and differences in the alimentary canal in many different vertebrates, specifically reflecting their diet. Herbivores generally have much longer alimentary canals. Carnivores tend to have the shortest alimentary canal, and that is because plant materials just take longer to digest, and most herbivores have a fermentation chamber in their gut to help them digest and make use of the cellulose, especially, that's found in large amounts in vegetation. So here's two stomachs, one from a coyote and one from a marsupial, actually, a koala. So koalas eat mostly eucalyptus leaves, and canids are all carnivores. They will eat some plant material, though, occasionally, but carnivores tend to have very stretchy, expandable stomachs, can eat huge meals at one time, and then can also easily fast and go days at a time without eating and not have any trouble with that. They tend to have the shortest intestines, and our intestine, so here's a typical carnivore intestine. Our large intestine is a little bit longer than a typical carnivore, but shorter than even our closest primate relatives. And for herbivores, the small intestine and the large intestine tend to be longer, and then they have often some kind of a fermentation chamber in the gut, like a very enlarged cecum, or extra stomach compartments like a rumen for providing a place for these anaerobic fermenting bacteria to live. There are some interesting exceptions to this, though. For example, pandas, which are bears. Pandas are in the carnivore class. They're in the carnivore order. They're much more closely related to other omnivorous bears than they are to any herbivores, although they eat mostly plants today, and they have a longer intestine, but they don't have fermentation in their gut, which means that they have to consume an enormous amount of plant material to get enough calories because bears lack a fermenting gut. And we call this kind of relationship where both types of organisms that are in a symbiotic relationship where both benefit, that's called a mutualistic relationship. So there's three basic types of symbiotic relationships with organisms. Parasitic relationship means that the parasite benefits and the host is harmed. If it's a commensal relationship, that means that the commensal benefits, but the host is neither harmed or helped. And a mutualistic relationship, though, that means that both benefit. So we have gut bacteria, even though we don't have the bacteria that make B12 or that can do the anaerobes that can do fermentation of cellulose, we do have several hundred species of bacteria that live not just in our gut, but in every orifice of our body. On our skin, bacteria absolutely cover the surface of our bodies. We have several pounds of bacteria each. And the species that we have varies, and it's also a new field of study. So everything that these symbiotic bacteria are doing is we're just scratching the surface now with science of what these bacteria are doing. It has now been found fairly recently that your bacteria that are found in your gut and on your skin and in your ears and up your nose and everywhere, that they actually assist the immune system in figuring out which are the bad guys, which are the bad bacteria, and where are there cells that are infected with viruses. Because without these symbiotic bacteria, your immune system just kind of shrugs and doesn't know what to attack. So this is a huge area of research right now that we really just barely know anything about. But it's turning out that our gut bacteria benefit us in a lot of ways and interact with our immune system even in a lot of very interesting ways. They also, as I mentioned in the last video, make metabolic intermediates like butyric acid that actually feed the cells of the intestinal epithelial layer in the large intestine. And we call all these bacteria our microbiome because we really are an ecosystem for all of these bacteria. And one of the things that has been discovered is that each person's microbiome, exactly which species are present, varies a bit from person to person. It even varies within people in the same household. It varies based on what kind of diseases that individual person has. So people who have type 2 diabetes tend to have a different grouping of species in their microbiome than somebody who does not have metabolic syndrome or type 2 diabetes. And there have been some experiments with fecal transplantation where they clear out somebody's microbiome because they have C. diff or some other infection. Usually it's for C. diff. They give very strong antibiotics to kind of wipe out the C. diff along with all of the healthy bacteria, and then they give that person a fecal sample from another person in a pill form, in a capsule, to reintroduce healthy bacteria into their gut. And it does actually work. I actually had an aunt who had C. diff in the hospital, and she was very close to dying from C. diff. And so they tried this experimental treatment on her where they cleared out her microbiome and gave her a fecal sample from a healthy person, which they often use kids for this because kids have the most diverse microbiome. And it worked. Within days, she was much, much, much better. We also know that surprisingly certain abnormalities of the intestinal tract are linked to bacteria like H. pylori, which cause ulcers. So this is just to show you the species diversity. So formiculates, bacteroides, and actinobacteria These are different classes of bacteria, and this is one of the approaches to studying the microbiota, is just to take a sample and just do genetic sequencing and just figure out how many species are present, what groups do they belong to, and it's been found that our microbiota changes as we age. It changes based on diet. So for example, breastfed infants have a different microbiota than formula-fed infants. Individuals who've been on antibiotic treatment lose some of the bacterial species that are found in their gut. People who are obese versus normal weight tend to have slightly different gut microbiota, and it's even been shown that you can affect a person's weight by giving them a fecal transplant from somebody in the opposite category. Unfortunately, though, it's not marketable because the healthy bacteria that obese people lack don't form spores and couldn't be therefore commercially marketed. So it's a very interesting, very new field, and lots of new research coming out all the time, and we barely understand it. I just listened to a podcast with a microbiologist who this is his field of study, and he said we're just barely scratching the surface of this field, that we really don't know what we're doing yet with the microbiota. So the digestive system is regulated by the nervous system because, of course, there's muscles involved, starting at the mouth, and every step of the way there's either voluntary muscles or smooth muscles involved at every step of the way. And there's also hormones that are involved, and that's the endocrine system. So we'll talk about a few of those. There are hormones also that are made by the liver that are really important as part of our kind of an ancillary to our digestive system. And one of these is the control of, relates to the control of blood sugar and also the control of fat stores in the body. So if you remember from biochemistry, our short-term way of storing sugar, because glucose cannot be stored in any large quantity in cells. So instead, animals store it as glycogen. And the two main places in our body where we store glycogen is our liver and in our muscle cells. And the muscle cells mostly use it themselves for muscular work. The liver, however, the liver's glycogen stores is mostly used to keep your blood sugar stable. So if you don't eat for 24 hours and your blood sugar goes down, the liver will release sugar into the blood, glucose. When I say blood sugar, I only mean glucose. Fructose and sucrose are not what we're talking about. If there's excess sugar, it will be stored. There's only a limited amount of glycogen that can be stored. Once all the glycogen stores are full, muscle cells will not accept any more glucose. And liver cells as well start turning it into fat. And that fat ends up in the circulation as triglycerides, and that fat then ends up in fat cells, in the adipose cells, to be stored. So hormones are going to determine whether that fat in the adipose cells can be used. So if your insulin levels are high, that tells the fat cells they need to store fat, not release fat. So people with chronically high insulin levels, which we'll talk more about that's part of metabolic syndrome, those individuals have much more difficulty releasing fat from their fat cells. So as your blood sugar is being regulated when you're not eating, the liver will first break down all its glycogen. The muscle cells tend to use their glycogen for their own use, but it can also be released into the bloodstream if your blood sugar is really low. And then finally, fat will be used. So as the glycogen stores get expended, insulin in the blood is going down, down, down. Finally, it reaches a point where the fat cells can now release triglyceride into the bloodstream to be used for energy by other cells in the body. So glucose in the blood is one of the things that we maintain by a homeostatic mechanism. And we actually have two hormones that affect glucose. We only ever hear about insulin, but glucagon does the opposite of what insulin does. So insulin is released when glucose is going up, and it lowers glucose in the blood by causing cells to take up the glucose. And glucagon does the opposite. So let's say you're fasting for your colonoscopy. Glucose levels in the blood are going down. Glucagon will be released, and glucagon triggers the liver to break down glycogen and release it into the bloodstream. So the liver is the most important organ in your body for glucose homeostasis, because the liver can release its stored glycogen in the form of glucose. It can break it down into glucose and release it. It can take in excess glucose from the bloodstream and turn it into fat or store it as glycogen. And it also releases glucagon when blood glucose gets too low, or it triggers the release of glucagon, I should say. Okay, so your normal blood glucose is in this range. As low as 60 is considered perfectly fine, but this is like average blood glucose level. Now when you eat a meal, it goes up above this, but not a huge amount, because insulin is then released from the pancreas. The beta cells of the pancreas release insulin, and this triggers the liver to take up blood glucose and turn it into glycogen. If it has enough glycogen already, it will turn it into fat, and it triggers other cells in the body to take up glucose. And then that should lower blood glucose back to the normal level. If there's no food coming in or just no carbohydrates coming in, glucose level goes down. Alpha cells in the pancreas then release glucagon, and this will trigger the liver to release glucose into the blood in the form from broken down glycogen. And eventually the liver can actually make glucose in a process called gluconeogenesis. So if you have fasted for multiple days, or you have a really low carbohydrate diet, the liver can make glucose. So if you remember back from our essential nutrients section, there are no essential carbohydrates. Carbohydrates are not essential nutrients because we can make them. So your liver can make glucose from fat and from actually protein breakdown products. Some amino acids can also be part of that process of making glucose. So we don't actually need to eat any glucose to have a stable blood glucose level. And that's what happens, if you have surgery and you have to fast for multiple days, your blood glucose level will be fine because your liver is going to use up all of its glycogen. Then when that runs out, it's going to start making glucose from scratch by the process of gluconeogenesis. And here's a little video just summarizing that interaction of insulin and glucagon, but I'm not going to show it to you right now, but you can watch it on your own from the PowerPoint. So insulin acts, almost all the cells in the body have insulin receptors, and when the insulin receptor binds, what's supposed to happen is glucose is going to be removed from the bloodstream and taken up by cells. But cells have a limit of the amount of glucose that they can take up. And once the cell has all of the glucose that it can take, it stops responding to the insulin. And that's what we call insulin resistance. Brain cells can take up glucose whether or not there's insulin present, but most cells require at least a little bit of insulin, especially fat cells. Fat cells will not take up glucose from the bloodstream and turn it into fat unless there's insulin present. This is why type 1 diabetics who have, when they're initially diagnosed, generally have zero insulin production. They tend to be losing weight dramatically because most of the cells in their body are unable to take up all that even though they have a massive amount of glucose in their bloodstream, it is not available to the cells in their body because there's no insulin present to tell the cells to take it up from the bloodstream. So glucagon and insulin, both produced by the pancreas, and they kind of do the opposite function. One raises blood glucose, one lowers blood glucose. So diabetes, or diabetes mellitus, this is from the root for honey, because it has been known for hundreds of years that this major symptom of diabetes is sugar in the urine, and doctors used to taste people's urine to find out if it was sweet to make this diagnosis. Luckily, we don't have to do that anymore. So there's two main types of this type of diabetes, where there's other kinds of diabetes as well that don't have anything to do with sugar in the blood. But diabetes mellitus is when you have excess glucose in the bloodstream. And there's two different reasons why we could have excess glucose in the bloodstream. One is if you have type 1 diabetes. This used to be called childhood diabetes, but because it usually does appear during childhood, but we now know that it can appear in adults, even older adults. We now know it's an autoimmune disorder where the immune system attacks and kills the beta cells of the pancreas. And when it's first diagnosed, often the beta cells are still trying. They're stem cells that produce the beta cells, and they're often still trying, and there might be a very small number of living beta cells that are still trying to produce insulin. There's ways to detect that now, and one of the new treatments for freshly diagnosed type 1 diabetics is the same kind of drugs they give for other autoimmune disorders, trying to stop the body from killing off all the beta cells so that some insulin production can be maintained. So this type 1 diabetics must inject insulin either by individual shots or using an insulin pump. And they're going to do that for the rest of their life unless we have some kind of a pancreas transplant or beta cell transplant that becomes available in the future, which there's actually people working on that. And I believe they've actually got a few experimental trials going on with giving type 1 diabetics transplanted beta cells to see if they can recover some of their insulin production. But this is a deadly disease without treatment. Children who got type 1 diabetes had weeks to live previous to the discovery and the production of insulin. Type 2 diabetes is completely different, and type 2 diabetes, individuals with type 2 diabetes, and there's multiple types. I believe now type 2 diabetes is broken down into 6 different subtypes that are characterized by slightly different issues with insulin production. But generally, type 2 diabetes is characterized by very high insulin levels. So the problem isn't there isn't enough insulin. The problem is that there's so much insulin and circulating glucose that the cells are simply not responding to it anymore. And this is called insulin resistance. Although now we know it's not because the cells aren't actually able to respond to insulin anymore. It's because they're full. The cells have as much carbohydrate as they can take, and that is why they're not responding. This is part of metabolic syndrome. There's a whole group of different ailments that go along with metabolic syndrome, and it's now known that they are all characterized by hyperinsulinemia. This is considered a lifestyle disease. It's a disease that's driven by, they're still working it out. It's not entirely clear, but seems to be mostly driven by the excess consumption of carbohydrates. And reducing carbohydrate in the diet is a classic part of the treatment of type 2 diabetics. So it's not always associated with obesity. That's kind of a mischaracterization. That metabolic syndrome can also lead to excess body weight. But about 30% of type 2 diabetics are normal BMI and aren't visibly overweight at all, but they do have really high insulin levels. So the new hypothesis is that each individual has a limit to how much fat they can store on their body. And you could reach that limit at a fairly low weight and still be normal BMI, or you might have to get up to 500 or 600 pounds before you reach your body's limit for fat storage. Some people can multiply their fat cells very easily. Other people, their fat cells are very reluctant to divide and make more fat cells. And that could be part of the difference. This is an area of very intense research right now because the treatment for type 2 diabetes has drastically changed in the last 10 years. It used to be thought that type 2 diabetics just needed more insulin. It was not known that this was a disease of abnormally high insulin. So it was thought, well, just like type 1 diabetes, we'll just give them insulin and that'll fix it. Or we'll give them drugs that will make them secrete more insulin. And that turned out that does not work at all. That will temporarily lower people's blood glucose, but they'll just get worse. And so people, type 2 diabetics who get insulin typically need more and more and more and more insulin until they're getting 10 times the dose that a typical type 1 diabetic would get. So the problem is cells aren't responding, blood glucose is too high. You can lower blood glucose also with diet, and that's become a very important intervention for type 2 diabetics. New treatments like metformin, which has replaced the initial treatments that they used to give type 2 diabetics. Metformin works by stopping the liver from making glucose. So it lowers blood glucose by a completely different mechanism than insulin. It stops the liver from pumping glucose out into the circulation. Type 2 diabetes used to be called adult onset because prior to the 1990s, it was never seen in children, and I mean not at all. And starting in the 1990s, we started to see kids with type 2 diabetes. And now the children as young as 5 years old have been diagnosed with type 2 diabetes. So this is an indication that our diets have radically changed. This is a disease that's caused by diet. Type 2 diabetics were extraordinarily rare 100 years ago. So that's too quick for it to be genetics. That means that it's diet. And the diets have changed. Here's another little video on diabetes. I'm not going to show it to you here, but it's in the PowerPoint. You can watch it, but it's pretty much just a summary of what I just said. The regulation of appetite is also involving hormones, and we already know some of these hormones that are involved, and some of them are very newly discovered, and they're like peptide YY is fairly newly discovered. We do know that insulin is a part of this process. High insulin levels, we now know the way chronically high insulin causes obesity. It is because it blocks fat cells from releasing their fat. And it causes them to store fat but not release fat. So insulin has to come down before fatty acids will be released from the adipose tissue. And chronically high insulin levels is now considered the basal cause of metabolic syndrome, which metabolic syndrome has been talked about for over 20 years. It's a cluster of things, including type 2 diabetes and obesity. But we now know that individuals with chronically high insulin levels also are at increased risk for all kinds of cancers, for heart disease, and for strokes and early death. So this is a very important issue facing our society today. The calories in, calories out hypothesis, which has been around for maybe since the 1970s, has been a total failure because it failed to take into account all of these hormones that control fat storage and that control absorption of nutrients. So we now know from many, many, many studies that long-term low-calorie diets reduce basal metabolic rate, which you remember is the resting rate at which your body burns energy, and they only result in temporary weight loss. And this is exemplified by a study that was done on the participants in the Biggest Loser TV show, that although everybody lost weight, they all also lowered their basal metabolic rate, and when they did 5-year follow-ups, almost everybody gained all the weight back or more. So that doesn't work. Another hormone which we didn't know about 10 years ago, which appears to be a really important part of this, is ghrelin. So when you go on a low-calorie diet to reduce the calories in, ghrelin will gradually increase and increase and increase, and eventually you're going to go off of your diet, because ghrelin, it signals your brain to eat, and you can only use willpower to overcome that for so long. So we now know, this has been much better understood just in the last 10 to 15 years, that fat storage and appetite are under the direction of hormones, and just doing the calories in, calories out formula is only giving you a tiny part of the picture of what's happening. So the hunger and satiety hormone. So satiety means feeling satisfied or full. So that's the opposite of hunger. Satiety and hunger are opposites. So we do know that there is a region of the brain that is affected that tells you eat. And one of the hormones that triggers that part of the brain is a hormone called ghrelin. Ghrelin is secreted by the stomach when it's empty. So if your stomach is empty, you will secrete ghrelin, and the level of this hormone can change over time. So somebody who's on a low-calorie diet, it has now been shown that their blood levels of ghrelin when their stomach is empty increase over time, and increase the signal to the brain to eat, and triggers a feeling of being hungry. So that's why it's so hard to stick to a low-calorie diet for more than about 2 weeks. That appears to be the limit for most people when they'll go off the diet, because ghrelin is increasing. 2 other hormones are insulin, which we already talked about, and peptide YY is a newly discovered hormone. Peptide YY, which of course is a peptide hormone, is secreted by the small intestine when you've eaten a meal. And that goes to your brain and says, I'm good, I have food. And peptide YY is increased by the consumption of fat. So a fatty meal makes you feel more satisfied and suppresses your appetite more because of peptide YY. Leptin, which has been known about for longer, this hormone has been known about since the 80s. Leptin is produced by fat cells, and it also suppresses appetite. And it's not clear why in people who are overweight, leptin doesn't appear to do the same thing. It was thought that perhaps people who were overweight weren't producing enough leptin, but it appears that they do, that people who are overweight have plenty of leptin. They have high leptin, yet it's not suppressing appetite. And so it's thought that perhaps brains can become resistant to leptin in the same way that cells are becoming resistant to insulin. When insulin is chronically high, cells stop listening to it. When leptin is chronically high, it's thought now that perhaps brain cells aren't listening to it. And this is a hypothesis of why people who are overweight, even though they have high leptin, don't have a suppressed appetite. So these are there are additional hormones at play, but these are the major ones. And an interesting result from bariatric surgery, so probably, most of you know something about bariatric surgery because of shows like My 600-Pound Life and other shows about bariatric surgery, that one of the most common types of bariatric surgery that's proved to be very successful is removing the outside part here of the stomach and making the stomach into a long, skinny tube. And surprisingly, that surgery, making the stomach into a long, skinny tube, has been weirdly more successful than making the stomach even smaller with other kinds of surgery. And the reason they now think is because Ghrelin, that's the hormone that makes you hungry, is mostly produced here in the outside curve of the stomach. So when they take that out, they remove most of the cells that produce Ghrelin. So they drastically lower the amount of Ghrelin that that person produces. And that's probably why that kind of surgery has proved through studies to be the most successful kind of bariatric surgery, even more successful than more extreme surgeries that make the stomach even smaller and remove part of the small intestine, supposedly to lower the amount of nutrient absorption. Which was called the that kind of more extreme surgery was called the Roux-en-Y surgery, and that has been shown to be less effective than just removing this outside curve of the stomach. And it's because of Ghrelin. Because if you take away most of the Ghrelin, the person just doesn't feel as hungry, and they just, you know, lose their cravings. And that has been an interesting result from that. Okay, so in this chapter we talked about the difference between undernourishment, where you just don't have enough calories, and malnourishment, where you're missing essential nutrients. And we talked about what those essential nutrients are. There are essential amino acids, there are essential fatty acids, and there's vitamins and minerals. There aren't any essential carbohydrates, however. The 4 stages of food processing: ingestion, digestion, which includes mechanical and chemical digestion, absorption of the broken down nutrients, and then elimination, which we really didn't talk much about that. The function of each segment of the digestive system: where do the different kinds of digestion take place? Where does absorption take place? It's pretty much all in the small intestine, a little bit in the large intestine. And we talked a little bit about other vertebrates, especially ruminants and other herbivores that have a large cecum. And then we talked a little bit about the hormones, glucose homeostasis, metabolic syndrome, and the hormones that regulate hunger and satiety. So that's the end of this chapter.